Okay. Hello, everyone. My name is Yunfei, and I'm a second-year PhD candidate in CS department of Vanderbilt University. I feel really honored to give this presentation of our work titled ADEPT, Autoencoder with Differentially Expressed Genes and Imputation for Robust Spatial Transcriptomics Clustering. Okay, so what we will go through together today are listed here. So I will first briefly go over the motivation, research background, and related work. And I will then focus more on the ADEPT method and result analysis part. And finally, we will go over some future work and leave some time for Q&A session. So let's just start with the motivation and research background. So what is spatial transcriptomics? Spatial transcriptomics is a groundbreaking molecular profiling method that allows scientists to measure all the gene activity in a tissue sample and map where the activity is occurring. So basically, ST allows us conducting scRNA sequencing while also measuring the spatial context of cells within the tissue. So since 2020, this ST technology are changing the way we understand complex tissues. There are two main categories of methods of ST sequencing. The first category is performed via fluorescence-based in situ transcriptomics. The other category consists of a combination of spatial barcoding and next-generation sequencing. The tissue size can vary from a small section to whole organ sections and from model organisms. The number of genes counted can vary from tens to thousands or even the whole genome. And a spatial location may range from a whole tissue domain to a large 500 micrometer by 500 micrometer region of interest, or down to a single cell or even finer. From a high-level understanding, the difference between spatial transcriptomics, bulk RNA sequencing, and scRNA sequencing techniques could be taken in this way. So starting from the bulk RNA sequencing, it basically captures the general idea of gene expression, but it fails to restore any cellular or spatial resolution information. But when it comes to the single-cell RNA-seq, the distinct cell types could be identified with their specific gene expression profiles. But for now, as we have like the spatial transcriptomics, we could visualize everything with the tissue context. So combining with everything we have for now, we could just try to identify the functional tissue. And spatial capture technology powers the Visium spatial platform using spatially barcoded mRNA binding oligonucleotides here. So basically here we're showing like the protocol of 10x Visium ST technique. So basically there are two methods for how mRNA molecules get a spatial barcode. So the first one is fresh frozen, and the second one is FFPE. So in this figure, 10x Genomics show the protocol of processing fresh frozen tissues. Firstly, we will take a photograph of the tissue before permeabilization. We're dissolving the tissue. Then the tissue is fixed and permeabilized on the slide to release all the RNA segments, which binds to the adjacent capture probes, allowing for the capture of gene expression information. And for each of the small round area, they have like a location-specific barcode, which could be processed later to retrieve the coordinates of each RNA segment. And each slide could approximately have around 5,000 spots for sequencing. And in the end, the cDNA is then synthesized from captured RNA, and then sequencing libraries are prepared. So why are we doing the clustering here for the ST data? So I think this is an important first step for most of the ST researchers. We want to define like the spatially coherent regions in terms of expression data and location adjacency. This is essential for downstream analysis such as like cell type identification, tissue annotation, and spatial variable gene identification or gene ontology analysis. So in recent years, several popular methods, including Bayes space, spatial GCN, CCST, and StateGate, have been proposed and demonstrated their superiority compared to previous baseline models. These tools use different mathematical or deep learning models to handle both gene expression, spatial context, or image information to perform clustering. But however, they produce results which are somewhat unstable and of great variances across different datasets. And here goes our motivation. To build a robust clustering with low variance, one big motivation for us is to understand the statistical properties of the excessive zero values in ST data. Just like what happens in the SARA-seq data, our ST data also has this kind of phenomenon with like excessive zero values in different datasets, which could be caused by either technical issue or other things like the noises and so on. However, some of them are true zeros. We refer this kind of excessive zero phenomenon as the dropout events here. And none of the aforementioned existing clustering methods design a specific or general approach to tackle the data quality effect across different types of ST data. So we were thus motivated to develop a robust clustering algorithm to deal with this kind of problem. And to deal with this kind of dropout events, to control dataset quality and also harness spatial context and transcriptional profile in ST data, we developed a graph-based multi-stage framework for robust clustering called ADEPT here. So basically, our method relies on the selection of differentially expressed genes and imputation of the multiple DEG-based matrices for the initial and also the final clustering of the graph autoencoder backbone, just to minimize the variance of the clustering results. And we're gonna introduce the method next. So here we show the overview of our method ADEPT. And the input data for the ADEPT is pretty straightforward. It's just the ST data. We have the expression profile, we have the coordinates, and we have the image as an optional input. And basically, our method has three main components. The first one is the data pre-processing component. ADEPT first constructs a k-nearest neighbor graph based on the spatial context. To construct the graph, a data quality control step here is integrated to remove the low quality genes. And the graph model input is then fed into a graph autoencoder with an attention mechanism. And this is just the second component. And the main goal for this component is to learn low-dimensional latent representation with both spatial context and expression information via graph autoencoder. And finally, based on embeddings ADEPT can use for the final clustering, we can just perform several different like downstream analysis. And we will go through some like technical details for each component next. And in the first data preprocessing component, the input ST data has expression profile, which is basically just a gene spot to gene expression matrix. We have spatial context, which is basically just like the spatial coordinates for each spot, and then optical image. In ST data, spots that are outside the main tissue area and the spots without any annotation are just removed firstly. The reason for doing this is because all those outlier spots might have like extremely low sequencing quality and do not have any adjacent neighbors. So we're just trying to remove all these kind of noises to maintain like a good quality for our input data. Raw gene expression data are then log transformed and normalized as just in the standard ST data processing protocol. And ADEPT uses an empirically determined non-zero rate of 0.14 as a threshold to screen out low-quality genes when the total count across cells of the gene does not meet a specific minimum number. So the number of the removed low-quality genes varied a bit in different datasets. Finally, in this component, the graph structure is built with the k-nearest neighbor algorithm, and we will be fed into the graph autoencoder next. For the second component here of ADEPT, it is just the main backbone of our method. It includes a graph attention autoencoder, and which consists of three core parts: the encoder, the decoder, and the graph attention layer. So ADEPT learns low-dimensional embeddings for all spots and perform an initial clustering based on them. And after we have done some refining work with the differentially expressed genes selection and the imputation afterwards, we're gonna have like another final clustering to have the final output of this method. And ADEPT selects differentially expressed genes based on the initial clustering results, and across different types of datasets, we use the same range of non-zero rates to control the data quality. And we're gonna select like a different but wide range of digit lists for each dataset. As for the like technical details here, so the encoder just consists of the input for the encoder just consists of the normalized gene expression profile and the edge lists. And for each node, the encoder generates node embeddings by aggregating information from all neighbors based on this equation 1 here. And we denote HU0 as the normalized expression feature of spot U. And by treating expression profiles as the initial spot embedding here, the Lth encoder layer generates the embedding of spot U in layer L as follows. So basically, it's just what the equation 1 describes here. And AUVL is the attention coefficient, which is gonna be acquired through the second and third equation of the attention mechanism. And NU just denotes all the neighbors of node U, including U itself. So this means we're just adding the self loops for each spot or each node. And the attention coefficient alpha UVL of layer L for every node pair UV in the encoder is just used to measure the importance of the neighboring node V towards learning a higher quality representation of node U on graph G. We're trying to introduce this kind of mechanism because like this attention mechanism is gonna make the final clustering result of our ST data pretty smooth. So basically this kind of attention mechanism could aggregate information from like neighboring cells to smooth out the final result. And I'll just show you some like more straightforward results, visualization stuffs later. And the output of the Lth layer encoder HUL here is just the final embedding, which we're gonna try to perform like the clustering with. It is just represents the hidden embedding with the lowest dimension, and the decoder attempts to reconstruct the normalized expression profile for each spot U, given the latent embedding of the encoder. More specifically, the output of the encoder is given as the input to an L layer, with the Lth layer of the encoder defined here as in the equation 3. And at this stage, for each dataset, ADEPT has selected several DEG lists based on the non-zero rates and extracted multiple DEG-based matrices. The strategy of our imputation method is to use the average expression value of the gene within the same cluster and the pseudo-labels that are obtained from our initial clustering results to complete the dropout entries. So here, what we do is to use like the information from all those entries with values to kind of like fill out some like pseudo values to know those zero entries so that the final clustering could be more stable. And in the final step of imputation, ADEPT just merges multiple imputed matrices to a final imputed matrix. It is then used as an input into the graph autoencoder for the final clustering. And in this way, ADEPT minimizes the variance of the final clustering result. And in order to evaluate the performance of our spatial clustering method, we used ADEPT to perform several downstream analyses on several annotated benchmark datasets, along with other 5 state-of-the-art tools. And now I will go through the experimental result details here. So basically, we're just using some like pretty famous and widespread dataset. So we're using the LPFC data here, which has 12 sections, and another human breast cancer dataset, which has only 1 section with the annotation, and a mouse visual cortex. And this one is produced using the StarMap protocol. And the first 2 are just using the TNS vision technique. And for the software and algorithm here, we're just using the ScanPy package for the overall processing pipeline of our data. And then we're just comparing with the state gate all the way to Bayes space with our method ADEPT. And here we just show the visualization and the table for our ARI results. So adjusted rand index ARI here is one of the widely used metrics for validating clustering performance. And here we show the ARI values for 10 sections of 3 different datasets we've been using for evaluation. Subfigure A and B showed the ARI values for 8 sections of the DLPFC dataset, and subfigure C and D just show the ARI values for the star map dataset and the breast cancer dataset. So for each dataset, we just try to run like 20 iterations for the clustering experiments. And in the box plot, the 5 horizontal lines just denote the from the bottom to top, just denote the minimum, lower quartile, median, upper quartile, and maximum value of our 20 iterations result. So the scattered points outside the box plot are outliers, which refer to either extremely high or low values in our experiment. And in this table, we can safely say that the highest average ARI in each column is highlighted, and we can see that for the result of ADEPT, we basically achieved like the best for 8 datasets out of 10. And one thing that is pretty interesting is that for the spatial GCN result on breast cancer dataset here, we can never like reproduce this kind of high ARI for them afterwards. After we got this kind of 20 initial clustering results, but just to maintain fidelity, we're just keeping this kind of value here. But just to mention that the spatial GCN might not achieve this kind of good result. This is just like a pretty outlier. And except for the ARI results, we also use the purity score and focus model score FMS here to evaluate the clustering performance. This table shows the purity score and FMS score comparisons between ADEPT and 5 other popular methods across benchmark datasets. And the highest average ARI and FMS score in each column are highlighted and consistent with what we observed in previous ARI table. ADEPT achieved the best performance in 8 out of 10 datasets for the purity score and 9 out of 10 datasets for the FMS score. And in this slide, we have the visualization result of our method. So subfigure A and B just shows the visualization of the DLPFC datasets, and C and D shows the breast cancer dataset and star map dataset. The left figure is our is the visualization of our prediction, and the right side is the manual annotation for each dataset. We just took the manual annotations as the ground truth to do the ARI calculation. We can say that ADEPT robustly recovers the layered patterns of cerebral cortical tissue and captures the cluster distribution of the tumor tissues in breast cancer data. And this one just shows the best clustering results from 6 tools on DLPFC slice 15, 16, 75. And each visualization result has 7 different clusters corresponding to 6 different cortical layers in white matter as suggested by the ground truth. Sorry. The rest visualization, except for our ADEPT, either has a non-smooth prediction results or fail to exhibit this kind of layer structures for the DLPFC tissue, but it is still open to discuss if the annotation itself is not good enough or the clustering algorithms have space to improve. Since the thinner layers here, like the red one or the orange one in the ground truth figure, is always hard to be captured or predicted accurately here. And as for the downstream analysis, we have performed analysis just to show the comparison of raw gene heat map and the imputed gene heat map of some layer marker genes from the previously biological studies with this kind of evidence. And the imputation module of ADEPT enhances the spatial patterns of these 6-layer marker genes. We, in subfigure B, which are heat maps, show the comparison before and after imputation of some chosen layer marker genes. And let's just take this kind of LAMP5 gene here as an example. So the raw heat map of the LAMP5 expression is illustrated in this kind of very scattered manner without any obvious layered patterns. However, after aggregating information from other gene channels, also with our imputation ideas here, you can see that the refined version of LAMP5 gene expression heat map is smoothed out with a nicely plotted contour along the ground truth regions of layer 2 in the ground truth. Meanwhile, in subfigure C, we could clearly see that the volume plot for LAMP5 reaches the highest overall expression in layer 2, just as the orange one suggested in the figure. So this kind of finding just aligns with both the previous biological evidence and also our imputation results. We can see that the ADEPT imputes gene expressions of biomarker genes for better spatial expression patterns. And in the end, we just use the UMAP visualization and PAGA graphs to infer the spatial trajectory of each cluster. We perform this analysis for clustering results generated by ADEPT, StateGate, and SEDR. The other 3 tools are not applicable to this kind of analysis, so we just skip them. And both ADEPT and StateGate achieved a pretty good spatial trajectory inference result here. But I mean, comparably, the ADEPT is slightly better because it basically eliminates some like weak connections between the layer 3 nodes and layer 1 nodes as compared in StateGate's PAGA graph. Okay, so in conclusion, we just developed a graph-based clustering tool for spatial transcriptomics data, which is called ADEPT. And we select like differentially expressed genes to perform imputation. And we can achieve the pretty robust clustering results with low variance. And we also improve the accuracy over a range of existing methods. And as for the future work, I think we're just gonna try to make the ADEPT scalable to big data sets, because for now, like one of the biggest weakness of ADEPT is that it has some CPU processing modules and GPU processing modules, and the transferring data between these two kind of mechanism is pretty hard. So basically, if you have more genes or more like spots in your ST data, the time consuming for this kind of algorithm running is going to be exponentially expensive. So we're trying to make this kind of pipeline more scalable to big data sets. And the second motivation is something we're trying to do now, is that we're going to try to train like multi-sections in the same SD data jointly to further increase the performance of our algorithm. So I would really love to thank Professor Maizie and Tyler Derr for their like supervising and instructions in this kind of like project. I have been struggling for like almost a year on this kind of project without getting anything that is progressive. But I mean, after the hard work and the instructions from professors, and also like the cooperation from all my co-authors, colleagues in this paper, we finally got something that is worth publishing. So I would really love to thank them for their contribution to this work. And I think that comes to the end of my presentation, and thank you for the devotion into this kind of presentation. And any questions?